So we're very pleased today to have with us Shuibing Chen and Steve Parker talking through their recent paper. The title of the talk is Integrative Single-Cell Multi-omics Profiling of Human Pancreatic Islets Identifies T1D-associated Genes and Regulatory Signals. Little bit of a bio for each of them. Dr. Shuibing Chen is the Kiltz Family Professor and Vice Chair of Innovation at the Department of Surgery, as well as the Director of the Center for Genomic Health at Weill Cornell Medicine, and her research focuses on leveraging human pluripotent stem cell-derived organoids for disease modeling and drug discovery. Her lab's work has been published in leading journals including Nature, Nature Medicine, Nature Cell Biology, Cell, Stem Cell, and Cell Metabolism. She leads multiple NIH data service programs, science programs, including DKNet, PancBase, and Multi-1D. She currently serves on the board of directors of both the ISSCR and International Chemical Biology Society. And her outstanding contributions have been recognized with numerous prestigious awards, including the New York Stem Cell Foundation Robertson Investigator Award, the ADA Innovation Award, and the NIH Director's New Innovator Award. So we're very pleased to have her with us. And Dr. Steve Parker, the Parker Lab generates multiple high throughput data sets on genome, epigenome, and transcriptome across species and disease-relevant tissues and cells at single-cell multi-omic resolution, and uses machine learning computational approaches to integrate and analyze their data. They aim to better understand the effects of genetic variation on chromatin architecture and transcriptional regulation at single-cell resolution. And the major goal of their lab is to generate mechanistic knowledge about how disease susceptibility is encoded in the non-coding portion of the genome from GWAS, with a focus on complex metabolic diseases, including diabetes, especially type 1 diabetes and related traits. I'm going to drop. He also comes to us from the University of Michigan. And I appreciate having you both here. And sorry, letting more people in. I'm not sure who's going to start first.
So I will share the screen. Okay, I will get started. Monica, thank you for inviting us. Also, thank you for very generous introduction. It's really my great honor today to present a joint program we ran together with Dr. Parker for several years, and still continuing, and some update from the progress. And just a little bit background, this project was funded by Human Islet Research Network here, and original motivation, we try to understand the biology, particularly how genetic factor contribute type 1 diabetes progression. And it's a large collaborative work between my lab, Parker's lab, Colin's lab at NIH, and Dr. Chen Liu's lab at Penn. And you will hear this collaborative work many, many times during this talk. And for this audience, I don't think we need too much introduction for the type 1 diabetes. Type 1 diabetes is largely considered as an autoimmune disease. People still believe the genetic factor play a very, very important role for the predisposition of the type 1 diabetes. And the GWAS has identified more than 50. I think the number keeps increasing now, maybe more than 100 signals that are related with the T1D progression. And of course, the T cell and dendritic cell play a very important role in T1D progression. However, study also showed the beta cells, some damage of the beta cell may contribute to the initiation of this process. They might provide some pre-apoptotic signal that can trigger the immune response. That's how this project was built on. And our for this particular project, it's really focused on two layers of the data, as Steve mentioned later, is both transcriptome data as well as epigenomics data. And as we mentioned at the beginning, it's a large collaborative project. As you can see, there are two major parts. And the first part is we perform single-cell RNA-seq ATAC-seq to single-nuclear ATAC-seq to the human islets exposed to cytokine-treated condition or Coxsackie virus B-treated condition to mimic type 1 diabetes progression, and run the paired single-cell RNA-seq single-nuclear ATAC-seq, and then to identify the genetic locus or even genetic variants that may contribute to beta cell damage And in MATLAB, then we take all these hits, we identify to biologically validated by doing the gene knockout, region knockout, and even SNP knock-in. So I will ask Steve to pick up from here to introduce all our sequencing and genetic work.
Yeah, thanks, Shuibing. Can you hear me?
Yes.
Okay, wonderful. So I guess first off, at a high level, thanks to Monica and the Sugar Science for the opportunity to share this work. It's really wonderful to interact with you all. And this has been just a fantastic collaboration with the Chen Lab. We've been working on this for a few years now. The paper finally came out after everyone knows a very long peer review process. And we're very fortunate to be able to continue this work within the HERN. So I'm excited about that. The slide, so I'm going to try to do this without sharing slides on my end. I can't really point to anything, so I'll try to be especially verbose, but if anything is unclear, just stop and interrupt me, and I'm happy to clarify things. But the slideshow now is a very high-level overview of the kind of outline of this study. So islets were isolated. They went to the Chen Lab at Weill Cornell, 8 different individuals in total. Some of those were profiled directly with single nucleus attack-seq and single cell RNA-seq. And then 3 of those individuals underwent a suite of different perturbations, right? Either control with omics, single nucleus omics profiling, a cytokine cocktail, or CVB4 infection. And those 2 perturbation conditions are designed to mimic an environmental exposure that could trigger type 1 diabetes. And so those are the experiments that we did. Shuibing's group did that, then sent us the samples. We generated the single nucleus attack-seq, single cell RNA-seq data, did all the computational integration, came up with some nominated targets at the end, and then I'm going to hand the presentation back off to Shuibing, who picked up on those nominated targets and did some more wet bench experimental validation of the targets. And that's kind of like a high-level overview. If you go to the next slide. Yeah, and so I just want to give shout outs to folks in my group that helped push this experiment forward. Cassie Robertson at the very top had some contributions to this, but really the person driving it was at the bottom, Ricardo Albanas, a former PhD trainee in the lab that stayed on and did a short postdoc to wrap up this work, and now is at WashU in St. Louis on the academic faculty track there. So Ricardo is wonderful. Most of the stuff I'm going to show is stuff that he did. Yeah, go ahead. Next slide. Thanks, Shuibing. So this is all the data combined together, 28 different sequencing libraries, 8 different individuals, basal and 2 different perturbation conditions. At the bottom, you can see the summary. There's about 120,000 total past QC high quality cells or nuclei. About 50,000 of those are attack and about 71,000 of those are RNA. And you can see the expected clusters here, right? On the left, you can see, alpha cells, beta cells, acinar, ductal, delta, immune, stellate, endothelial. And on the right, you can see the data split out by modality with the attack on top and the RNA on bottom. And you can see that the clusters are very similar, indicating that I think we've done a reasonable job at profiling at single cell or single nucleus resolution to distinct modalities on these samples. If you go to the next slide, just a little bit of standard kind of QC to convince you the data are of reasonable quality. Here on the left is the dot plot of the single cell RNA-seq data, where the y-axis lists the different cell types and the x-axis lists the different marker genes. And you can see, as expected, for example, in the top left corner in beta cells, insulin is very highly and very specifically expressed there. And you can see a nice diagonal of marker genes as you go across the alpha, delta, gamma, et cetera, cell types. On the right side of the slide is the single nucleus attack data. And what we've done is taken a similar approach where, again, the y-axis are the different cell types and the different columns represent a marker gene. So that first column is the insulin marker. You can see insulin gene at the very top of the gene track. And you can see the beta cells, of course, have kind of really high chromatin accessibility with the attack modality at the insulin locus. And then you can see kind of, if you take a step back, a diagonal going across the plot indicating high accessibility, very specifically in the cell types that express highly that marker gene. So hopefully this is enough of a big picture to convince you that the data are of reasonably high quality. If you go to the next slide, here's just another very high level view of the data where on the x-axis, we're listing each of the different samples that were run. And I'm going to kind of progress upwards in the stack of data right here. The next row above the sample labels indicates the treatment, where a white box is the baseline, a blue box is the cytokine perturbation condition, and the kind of pinkish magenta box is the CVB4 perturbation condition. At the bottom of the labels, I've bracketed groups of 3, where you can see those 3 different samples that got baseline and the 2 different perturbation conditions. The next stack up is the not the next slide, sorry. The next stack up just tells you the number of the proportion of islet cells that we see across the conditions, where we broke that up between alpha, beta, delta, and gamma, and then higher up, the top stacked bar plot is the total proportion of all cell types. And you can see kind of on the right-hand side of the plot, these were samples that just came in with overall lower islet purity. So you can see a higher representation of non-endocrine tissue in those specific samples. But once you take a glance down and look at the islet cell types, you can see they're all kind of like getting reasonably well aligned with representative proportions. So the next thing we wanted to do is do differential expression across these conditions. And we focused first on the RNA modality, trying to figure out what is the genes and the pathways that are changing when you introduce this cytokine or this CVB4 proximal environment to the cells. If you go to the next slide, this is the kind of pathway analysis of that, where the x-axis are the different Go terms that we see as significantly enriched. The top plot represents the cytokine infections, and you can see the red bars. These are Go terms that reach a 5% false discovery rate, and they represent things that kind of are what you would expect. Response to stimulus, basically responding to that cytokine perturbation environment. And then the bottom bar plot is the CVB4 infection bar plot, and you can see the red bars in the middle of that, indicating different Go terms are coming up, but still clearly the cells and nuclei are responding to that infection, but in a slightly different way that they respond to the cytokine perturbations. We had a bigger question here, and that was, how well do these laboratory environmental conditions mimic actual conditions that might be happening inside human samples? And so the next slide, what we did is a differential analysis comparing an orthogonal data set from HPAP, individuals that are autoantibody positive on the x-axis versus normal controls. And what we're plotting on the x-axis here are the differential expressed genes, log 2-fold change of autoantibody positive versus healthy controls. And then the y-axis, we're plotting the differential expressed gene log-fold change versus baseline. The left plot is CVB4, and the right plot is the cytokines. And the take-home message here is that you see most of the points are kind of along the diagonal that would create a positive slope. And the Spearman correlation for CVB4 infection is 0.22 with a p-value of 0.038. And for the cytokines, the Spearman correlation is 0.39 with a p-value of 10 to the minus 12. So in general, we think the kind of genes and pathways are going in a similar direction in this environmental perturbation condition as they are under like in vivo autoantibody positive conditions in a human. So we think that these are reasonably good models to kind of recapitulate T1D environmental exposures. If you go on to the next slide. Now I'm going to shift from talking about the RNA modality to talking about the epigenome or the chromatin landscape. And on the left is a cartoon that depicts how we use an information theory approach to quantify changes in the chromatin landscape. And this is based off of a paper on the bottom right that we published back in 2021, where Ricardo Albunas was the lead author. And again, this is just a methods paper of how we use information theory to quantify changes in the chromatin landscape. And then on the left, the cartoon kind of quantifies this, where in the top panel, you see a cartoon where transcription factor is bound in the middle, and that transcription factor binding induces nucleosome phasing adjacent to the bound transcription factor to the left and the right. You can see the DNA wrapped around the nucleosome, but the like overall open accessible region in the middle where the TF is bound. So you can think of this cartoon plot the x-axis represents kind of distance away from where that TF is bound, going to the left upstream, to the right downstream, if you think of things that way. The y-axis of this cartoon represents fragment size in attack-seek data. When you do an attack-seek experiment, you get this diversity of fragment sizes from really short fragments to kind of longer fragments. And that fragment size actually encodes important information about the chromatin landscape, where you can see towards the bottom of the plot, there's these blue points to the left of the transcription factor. They're small fragments, right, that happen between the TF and the phase nucleosome. And to the right, there's black points that, again, small fragments between the TF and the phase nucleosome. In the middle, there's these red fragments, and those are fragments that span the bound nucleosome, right, where TN5 integrated on either side of the nucleosome. It's kind of this medium-sized fragment that you get. And then as you go up the plot, these are bigger and bigger fragments, right, where they either span the nucleosome, maybe they span two nucleosomes, maybe they span the TF and the nucleosome, and we can kind of understand all of that based on where the motif for the TF occurs, the size of the fragment that we see mapped in that region, and then we just take the midpoints of those fragment sizes and we plot them. So that's what those kind of colored circles are. And then at the bottom is kind of an abstract representation of how we use information theory to quantify the structure and the organization in what this is called a V-plot. So Steve Henikoff was the first person to make a V-plot. We didn't invent that. We just kind of applied information theory to quantify the structure in that V-plot. And it's based on how well a transcription factor organizes chromatin around where it binds. Okay, so hopefully that all makes decent sense. If we go to the next slide, here's actual data from the study. The top row represents beta cell chromatin profiles, and the bottom row represents the immune cells. And the immune cells are really, really low, abundant in these samples, so not a lot of data. But on the left column, you can see CTCF, and you can see in that real data, you can see the pattern emerging in that V-plot. And at the bottom of the V-plot is where we've quantified the information content in the V-plot. CTCF happens to have this really long residence time on chromatin. It sits on chromatin like a boat anchor, and it induces this very well canonical structure, recognizable structure. And you can see CTCF is very well positioned both in beta cells and immune cells, not surprisingly. If you go to the middle column, FOXA, the chromatin organization looks a little bit more structured and organized in the beta cells than it does in the immune cells. And that makes sense. FOXA is this really important transcription factor in beta cells. And you can see if you look at the information content plot, there's higher information content in beta cells than in the immune cells. And then on the right column, we're trying to show an example that's higher in the immune cells. This is SPE1. This is an important immune cell transcription factor, and you can see that in the immune cells, even though there's not a lot of them in the data, it's very noisy. It's clearly much higher structure in the immune cell population than in the beta cell population. So we're going to use this to kind of examine dynamics in the chromatin landscape and changes across the different cell types. If you go to the next slide. On the y-axis here, again, are the different cell types, starting with beta cells at the top. On the x-axis are different transcription factor motifs, and we looked at a collapsed set of 540 different motifs here. And then the color just represents a z-score for how we quantified the structure, the organization of that V-plot. And you can see over on the right, we've highlighted a few factors that are important in beta cells and islets. So RFX factors, FOX factors, HNF4. And you can see at the top of the heat map, there's more yellow there indicating more structure around the chromatin regions where those TFs bind. Whereas over on the left, you can see we've highlighted a few examples of these are really known important immune genes like IRF, GATA, SPE1 again. And you can see the yellow highlight really specifically in the immune cells and not in the other cell types. So clearly, the chromatin organization is quite different across these cell types, and you can see that in the single nucleus resolution attack-seek data. Now we were interested if chromatin dynamics change as a function of those perturbation conditions. If you go to the next slide, Shuibing. Yeah, so here we're showing those, again, V-plot information scores as a function of the perturbation conditions, and I'll focus on the beta cells on the left. Now I'm showing the x-axis are either baseline, cytokine, or CVB4 conditions. and the y-axis represents a few different transcription factors. And you can see, I want to highlight at the bottom of the heat map in the center, under the cytokine conditions, those IRF motifs, these are immune programs, are really getting activated. You can see it immediately in the chromatin landscape, right? Like those are not active under baseline conditions and really not happening much under CVB4, but under cytokine perturbation conditions, the chromatin landscape is really getting dramatically modified and much more well structured as a result of the activation of these transcription factors. And then there's examples on the immune cell side too that I won't highlight too much because this is really going to be more of a beta cell story in the paper that I'll hand off to Shuibing in a few more slides. But what we wanted to think about next is enrichment for these open chromatin regions for GWAS. And so again, the y-axis is different cell types. The x-axis here is the fold enrichment when we look at attack-seq peaks. And the three different columns, I'm showing you three different GWAS studies. And I'll focus the first one on type 1 diabetes GWAS. At the bottom, you can see the immune cell attack peaks are, at the bottom, you can see that red point. The immune cell attack peaks are highly significantly enriched to overlap the type 1 diabetes GWAS signals. So not surprising. But the cool thing is, if you look at the very top of the plot, beta cells are too. So some of those T1D GWAS signals, clearly the immune system is important, but this is showing beta cells are actually really important too. And then the other two diagrams are just kind of to convince maybe a skeptical reviewer that the data are of high quality. We see high enrichment for type 2 diabetes GWAS in the beta cell attack-seq peaks, and we also see high enrichment for fasting glucose GWAS in the beta cell attack-seq peak. So overall, things are looking good. The beta cell is looking important for type 1 diabetes genetic predisposition. If you go to the next slide, Shuibing, now we're starting to really try to figure out if we look at any given T1D locus, which is the x-axis in this heat map, there are 136 different T1D GWAS signals that we looked at the time of this publication. Which cell type do they look like they're probably operating through? And again, cell types are on the y-axis. The coloration here represents this analysis where we took, for any genetics aficionados out there, the posterior probability weighted GWAS signal for the variant, and then also the intensity of the attack signal in that region. And so you end up getting really red signals where it looks like the chromatin architecture is really important in that particular cell type at that particular GWAS locus. So if you go, Shuibing next animation, she was zooming in and showing this cluster of T1D GWAS signals look like the chromatin architecture is very active specifically in immune cells, right? Not surprising to see a lot of T1D GWAS loci looking like they're operating through the immune cell population. If you go to the next animation, over on the left, we thought this was a little bit more interesting because it was indicating that the chromatin landscape for those T1D GWAS signals are highly relevant to beta cells, or maybe like islet endocrine cells in general. And if you hit next animation, I think it'll highlight, here's a positive control. On the bottom left, you can see those vertical arrows pointing to insulin. I think it's very reasonable to expect that the insulin T1D GWAS locus is working through beta cells. So very nice to see that those insulin GWAS signals look very beta cell specific in this computational analysis we did of the chromatin architecture data. But the insulin locus is not the only locus there. And so over on the right, we decided to pick out a few other loci that looked very interesting from a beta cell perspective, and this kind of partitioning of context agnostic GWAS to maybe these are really operating through beta cells. So I'm going to walk you through a few more computational approaches we did to look at those loci, and then hand it back off to Shuibing to show the rigorous wet lab work that she and her team went in and did to validate these loci. So here's the TOX1 locus. And it's a little bit of a busy plot, but on the left, you can see the attack-seq tracks, again, for each of the different cell types. At the very top, you can see the type 1 diabetes variants. The red one is the lead SNP, and then all the other black ones are SNPs that are in close LD with the lead SNP there. And there's a kind of vertical rectangle that zooms in, and if you look on the right part of the slide, that rectangle expands to have a closer, further to the right, it expands to have a closer look where we've done genetic fine mapping and functional fine mapping, these statistical genetic approaches, to try to narrow down on which of those SNPs that we think are the functional SNPs and which region of chromatin accessibility do we think are the important ones. And so these are the regions that then Shuibing is going to take and do direct perturbations and more cellular assays on. So this is the TOX1 locus. Here's the locus called MEG3 DLK1, where you can see on the right side of the plot, there's these really interesting ataxic peaks. And under different conditions, CVB4 and cytokine conditions, you can see these potential loop formations that get stronger and stronger as you have these different environmental condition, the perturbations that are really hard to see under the baseline conditions. And then one more locus that we looked at, RAS GRP1, again, where on the right you can see the genetic fine mapping, functional fine mapping that takes us to potential effector cell type, potential effector regulatory element, and potential effector variant. And I think that's the last data slide that I'll show before I hand it off to Shuibing to tell you more about the downstream experiments.
Thank you, Steve. Thank you for very nice introduction about the bioinformatics part of the whole story. And that's actually more than 2 years work. And after that, we think we really have some clues or maybe some ways we might be able to prioritize or think about how to run validate these genetic factors. And we have been interested in this platform for a pretty long time, and starting from using the human pluripotent stem cell as a platform to understand how GWAS hits contribute to the disease progression. And we really started from type 2 diabetes. We knock down the genes, knock in SNPs. And for the type 1 diabetes, the first story we work on is a GLIS3 knockout with some genetic variant knock-in. And usually the way we run it, we first create isogenic human embryonic stem cell iPS cells and differentiate into pancreatic beta cells. And then we monitor their cell survival function in vitro. We can challenge them with disease conditions, monitor their survival function in vitro, and then we can even transplant in the mice. And to if we want to do some drug evaluations, we can put it in mice and then monitor the disease progression in vivo. And using this platform, we can analyze the genetic network, gene-environmental interaction, and of course, for drug discovery. And with this platform, so after we get the gene list and hit genetic variant list, and as well as the gene list from Parker's lab, and then Zheping and later Xiaoshan, two very talented postdocs who pick up this project. So we decide to use a three-way approach. As if we work on the wet lab, of course, genetic variant is very important, but knock-in is a very labor-intensive work. So we actually did it in the reverse case. We start from gene knockout, and if we see a phenotype, we move to the region knockout, and then we move to the SNP knock-in, and then we create this iPS cell line, carry the risk, non-risk allele, and differentiate into beta cell and monitor their survival. So that's the very beginning of the wet lab validation part. We create this in knockout insulin GLP. We basically create gene knockout on the insulin GLP reporter line. The purpose for that is it's very easy to monitor the differentiation. We can simply use the percentage of GLP as a readout. In the meanwhile, by focus on the GLP positive cell, we can analyze the cell survival only in the beta cell cluster. So this is some just sequencing plot to show we do have knockout lines for each individual genes. And when we work on the isogenic system, clone-to-clone variation is always a concern. So we always have at least 2 isogenic clones for each genotype to make sure it's not whatever phenotype we see is not because of clone-to-clone variation. On the right, you can see the Western blotting to validate the knockout of these genes in human pluripotent stem cells. And the first thing we monitor is their differentiation efficiency. This is an insulin GLP reporter line, and as the up view panel, you can see a simple flow cytometry analysis to monitor the insulin GLP signal. In the wild type, we have 2 clones, and we get usually around 20 to 30% differentiation efficiency. And in these 3 knockout lines, like DLK1, RAS GRP1, and also TOX knockout, we can see the differentiation efficiency got significantly decreased and at different levels. So this is the first phenotype we see. And since we are interested in type 1 diabetes, so beta cell survival is also another very important readout. And now we monitor the beta cell survival in the baseline level. And at the left, again, if you can see a flow cytometry, we use annexin 5 and DAP as a readout, and we focus on the bottom right panel, so which the annexin 5 positive, DAP negative, which label the early apoptotic cells. And in the wild type cluster, we see relatively low percentage of the early apoptotic cells, and in the DLK1 knockout or RAS-GAP knockout condition, we see a significant increase of the apoptosis. But in the tox knockout, we don't really see a big difference between wild type and knockout. So then we decide to focus on the DLK1 and the RAS-GAP, and this is another assay we use is immunostaining to stain for the cleaved K-space 1 positive cell as a readout for apoptosis, and we stain the insulin with green color, and we quantify the percentage of cleaved phase 3 positive cells in the GLP positive cells, and the right of the quantification, you can see we see a significant increase of the apoptosis cells in both DLK1 and RAS-GAP knockout lines, which is consistent with our annexin 5 flow cytometry data. So since we find these hits from T1D conditions, so the next thing we did is to challenge them with the cytokine condition to mimic the T1D. And now we see a major, even more dramatic difference between wild type and DLK1 knockout. Oh, sorry, I think there's something wrong with the image, but you have to trust what I'm saying. So now we see a significant increase of the cell apoptosis in the DLK1 knockout as well as RAS-GAP knockout lines in the cytokine-treated condition. So next, we know the gene knockout, we can see some phenotypes. The next question is whether it's related with the regulatory region or even the genetic variants we identified. So the second step we take is we call region knockout. We usually knock out around 200 to 500 base pair, depends on the values of the ataxic peak. And then on the upper panel, you see the PCR of the genomic DNA. We can clearly see it's a very clean knockout. Again, for the region knockout, we also have 2 isogenic lines for each genotype. And we want to know whether if we knock out this region, it's affect gene expression. And we isolate the RNA and then monitor the DLK1 or RAS-GRP1 expression. And indeed, in the knockout region, we see a significant decrease of the gene expression. And we do see a pretty strong clone-to-clone variation. So now hope you appreciate how important it is to have multiple clones in each genotype. And we use a similar assay as a gene knockout to see whether region knockout gave us similar results. And in this round of experiment, the direct differentiation variation is not as large as QRT-PCR. And here you can clearly see the phenotype in the region knockout, DLK1 region knockout, RAS-GRP1 region knockout. We see a significant decrease of the differentiation, and we get much less insulin GLP positive cells. And we also monitor the beta cell survival. Again, we use focus on the annexin 5 positive, DAP1 negative clusters in the insulin GLP positive cells, and in the DLK1 region knockout and RAS-GRP1 knockout region knockout beta cells, we see a significant increase of the apoptotic cells, which is further confirmed by the immunostaining using a cleaved K-space 1. I just won't go through it one more time. This time we also challenged with the cytokine conditions. And when we monitor the annexin 5 in GLP positive cell, we see a significant increase of the cell death. When we use a cleaved K-space 3 as a readout, we see very similar trend. So then we push to the last step is we want to study the genetic variant that has been nominated from this very extensive study. So when we study genetic variants, the first thing we need to do is to sequence with so-called wild type lines. because the definition for SNPs is more than 1% of population should carry one allele. So, and when we do a lot of knocking study, we realize many so-called wild-type lines, they are either homozygous or heterozygous for the risk allele. So that's usually the first step we do, but for this case, we are pretty lucky. The wild-type line, they are homozygous for the non-risk allele GG. So we use a very traditional Cas9 knocking approach to create two isogenic clone which carry the AA risk allele. And the first thing again, you can see the QRT-PCR is not very stable assay. We also see a large clone-to-clone variation, but when we monitor the QRT-PCR level DLK1 expression, we still see a significant decrease of DLK1 mRNA expression in the cells carry the risk AA allele. So we also monitor direct differentiation consistent with the gene knockout, region knockout. We see a slight decrease of the direct differentiation. And as you can see, most of the time, the phenotype in the SNP knocking is not as strong as the gene knockout or region knockout, which is kind of expected because we only lose part of the gene expression, not completely remove the gene. And we monitor the beta cell survival. At the left part, you see the annexin 5 to monitor the apoptotic cells. At the right part, you see the CleavK space 3 to label the apoptotic cells. Very consistently, they show that in the beta cells, insulin GLP positive cell carrying the AA risk allele, we see a significant increase of cell apoptosis. And lastly, we challenge them with a T1D condition. So again, consistently, even in the cytokine-treated condition, the cell carries the AA risk allele, they show increased apoptosis. So using this insulin GLP reporter line, we actually have the ability to purify the insulin positive cells, like beta-like cells, so that allow us to do some mechanism study. So this is very high-level mechanism study. We basically purify the insulin GLP positive cells derived from wild-type or DLK1 knockout lines, and we send them for the RNA-seq and ATAC-seq analysis. On the top, you see the PCA plot of the RNA-seq analysis, and bottom, you see the PCA plot of the ATAC-seq analysis, and the middle, you see the gain or loss of the peaks in the ATAC-seq analysis. And first, from the PCA plot, we can see the wild-type and knockout are clustered pretty separately, suggesting they do have a lot of major difference, and variation across different samples are not that big. And when we run the pathway analysis, as Steve mentioned before, and this time we find many pathway change in the DLK1 knockout condition, but I just want to bring you one of them, which is NOA, NO and ROS. And then we use this exactly same approach to run the RNA-seq of the insulin GLP positive cells isolated from wild-type and RAS GRP knockout beta cells, and run the RNA-seq ATAC-seq peak again. I just want to bring attention to the right part. Although they look very similar, they are not the same plot, but we still see ROS signal come out. So the next question will be, is ROS really important? If we can rescue the ROS signal, can we see the partially rescue the phenotype? And the answer is yes. And I know it's a busy slide. On the bottom up, you see the DLK1 knockout. Bottom is RAS GRP1 knockout. And we basically monitor the percentage of insulin GLP positive cells. And in the DLK1 knockout line, you see a significant lower of GLP positive cells. And when we add the NAC during the which is antioxidant reagents during the differentiation process, we can see the differentiation level go back to the level that is almost undistinguishable from wild-type lines. And we see the very consistent phenotype in the RSGLP knockout lines. And it looked like they share the same downstream, very bottom downstream mechanism. Then we want to know whether they share any genes if we check the DEG list. And very surprisingly, we only find 5 common genes from the long DEG list between the DLK1 knockout and RAS GRP1 knockout, although they have the share very common downstream mechanism. But what is interesting, we are still investigating, is that that is 4 out of these 5 genes, they actually has been associated with T1D, suggesting there may be some genetic network of the T1D effector genes or gene associated with type 1 diabetes identified from GWAS study. They interact and either regulate each others. So I think we will stop here, and then we we have one more slide, but this is a summary slide. And so basically we show you a story. We start from single cell RNA-seq, ATAC-seq. I know many labs right now running a lot of fancy sequencing technology, and we use that to run extensive genomic epigenetic studies to nominate the genetic regions and to correlate it with GWAS hits. And then we validate it in the wet lab using the gene knockout, region knockout, and SNP knock-in. And we've shown that biologically, the genes nominated from the epigenetic study can really affect beta cell differentiation and beta cell apoptosis. And I just want to acknowledge my lab member first, who work on the project, and then also our collaborator from Collins Lab and Penn, and Steve. I will leave the Parker's Lab acknowledgement for you.
Yeah, this has been a wonderful collaboration. Lots of folks contributed. Just want to highlight again the name, and I read at the bottom of the slide, Ricardo Albanus, who's now at WashU in St. Louis. He's been great. Thanks, Shuibing.
Yeah. And lastly, I'm very fortunate to participate in a large program funded by NIDDK, also her. So, and Steve is a lead PI of the project. Do you want to say something about PanBase?
Yeah, I just wanted to introduce PancBase to everyone, and also say thanks to Monica for reaching out. This D challenge that's coming up, you're gonna hear from PancBase folks. I think it's October 17th, if the date's
That's
right.
right.
But maybe,
Yes.
yeah, maybe Monica can say more. But this is basically a infusion of AI and machine learning and statistics to kind of aggregate and analyze all things pancreas and T1D related. And Monica has this really cool data science challenge around resources available here and HPAP and other parts of the community. So just wanted to start to raise awareness about this newly emerged program and answer any questions that folks might have. Thanks.
Thanks, Steve. We hope you join on maybe if you have time on the October 17th discussion with the participants in the D Challenge. We have a big group this year, and they're definitely, kind of diving into the PancBase and sniffing around the HPAP resources as well. And so it should be very exciting. I want to open up to questions now pertaining to this paper, which is just a huge amount of work and really beautifully presented by both of you. We have a bunch of people in the audience. If you'd like to raise your hand or just drop something in the chat, feel free. Hard just looking.
I see we have one question in the chat, Steve.
Yes, here we go. From Peter Thompson up in Manitoba. Very exciting study. I'm wondering whether you think the SNPs that affected beta cell apoptosis may also affect beta cell function. Also, what about imprinting at DLK1 and how that might affect its regulation in T1D?
Maybe I can pick up the first part about the beta cell function.
Yeah.
It's a great question. We are still working on that. The reason for that, as you can see, we see a pretty strong apoptosis phenotype. When we see a strong apoptosis phenotype, it make monitoring beta cell function a little bit challenging. The experiment to experiment variation is pretty large. But for now, I would say after 8 or 10 rounds of experiment, we do see a trend of decreased response to glucose stimulation, but we still need a further confirmation.
Nice.
Yeah, and maybe I'll ask about the imprinting aspect. So yeah, I'm aware there's reports about imprinting at this locus. I think the nice thing about the GWAS signal is I think that would be separate from the imprinting mechanism, and I haven't thought through very rigorously about how imprinting might affect it, but certainly if DLK1 is sensitive to genetic variation dosage that influences T1D predisposition, then imprinting would probably have an effect there as well, would be my guess. I don't know if you have any, if you want to discuss this further, I'd be happy to chat more about it. But yeah, it's a really good question. Thanks.
Okay, anyone else? Okay, while we're waiting, what percentage of T1D patients have these three T1D risk signals? And if it's not known, how can this be further ascertained?
Yeah, maybe I'll start with that. That's a great question. It is known. I don't know it off the top of my head. Usually the minor allele frequencies for these risk variants are generally above 5 to 10% in the population. So I don't know exactly the minor allele frequencies for these three different signals, but you could basically kind of use the population allele frequencies to get an expectation of how many have those three. Maybe you're kind of going in a direction of, is there a lower likelihood to see all three of those risk genotypes in an
Yeah,
individual?
that's another question that was a follow-up.
Yeah, I think you'd have to have a really the problem with T1D is pretty rare, so it's hard to get really large sample sizes to answer that in a really adequate way. I will say for T1D, folks have looked at this question and they don't see any evidence of deviation from expectation across loci. That maybe is different for T1D, although I think it's an open question because the sample size just doesn't allow for those type of more robust statistics that you can do with T2D.
Okay, yeah, so it was looked at in T2D, but still needs
Yeah,
more fill-in in
yeah,
T1.
yeah.
Okay, and then, among these sort of the genes and the regulatory elements you've nominated here, which ones do you believe could potentially be the most promising therapeutic targets?
Maybe I can start. So
Yeah.
it's a great question. I mean, we are always interested into develop drugs. I think developing direct drugs to target these regions can be potentially challenging.
Yeah.
Based on if you monitor drug capability for each of them, like even DLK1 or RAS-GRP, is not that straightforward. It's not the traditional drug target like a kinase or GPCR we are
Yeah.
talking about. But I think if I recall it correctly, there are some drug candidates that can potentially change the DLK1 expression in an indirect way. So that's potentially doable.
Yeah, maybe
Okay.
can I ask, like, or add to this? I agree with everything Shuibing said. Just thinking about this, like maybe at uh, a different higher level elevation, the observation is these independent genetic signals converged on a similar pathway, right? So
Mhm.
and then the other cool thing is like, well, you knock out the gene, you get a big effect. You knock out the regulatory element, you get a middling effect. You knock out the variant itself, you get this really small effect. All, like Shuibing was saying, this is kind of consistent with biological expectation. So maybe none of those exact things are the right target. But now that we've identified this like convergence in this pathway, there's another druggable target in the same pathway, right? And so I think that's where this kind of network biology that Shuibing was talking about becomes really an attractive axis of investigation to find, perhaps better druggable targets that maybe they're not GWAS hits, and maybe they're not GWAS hits because
Right.
you can't mess with those things at all. They're like super sensitive, right? So anyway, that's just adding on a little bit to what Shuibing said.
Are you guys going to continue the inquiry in a temporal fashion in terms of, which parts of the network go offline or online, in sequential fashion? Yeah. Okay. Seems
Yeah,
like.
absolutely. We're doing a lot
Yeah.
of network biology and science, building on kind of this publication's what, probably a couple of years old now, honestly. So
Right.
by the time things come out in peer review, but yeah,
Yeah. Yeah. I know. Ergo bioarchives and the
yeah, yeah,
preprint,
yeah,
the preprints. So
exactly.
anyways, here's a question from Yasi Agazada, I think from Montreal. Thank you very much for a nice presentation. Question for Dr. Chen. When the percent of beta cells decreased significantly, was it only due to apoptosis, or did you get more of endocrine progenitors, alpha delta cells, et cetera? Did you see any differences in islet architecture?
Oh, yeah, this is a wonderful question. It's just because of the time limitation, I did not get a chance to go through the detail of the data. For several knockout lines, we start to see because we do the knockout in the human ESL, so we start to see the differentiation defect even at very early stage at pancreatic progenitor stage, even before the endocrine progenitor. So I think it's better way to answer this question. Now we have a new inducible knockdown system, so we can add doxycycline to add DKS9 at a different stage of the differentiation, so we can study the stage-dependent effect. Islet architecture is it's a very good question. I think for ESL differentiation field, I will say the organoid we derive is not good enough or consistent enough to study the islet architecture yet. And we are kind of moving towards that goal to get a more high throughput automation system to make more consistent islet organoid so that we can study higher resolution difference.
Great. I've got Nicole Krentz if she wants to unmute herself and ask the question.
Hi, Steve. Hi, Shuibing. I guess I'll continue with the Canadian line of questioning. It's a very beautiful talk, and it's a great example of how the power of stem cells to disentangle some of these GWAS hits. I was curious about the regulatory variants and whether you have any hint as to whether there's a transcription factor that's common. Are you perturbing binding sites? All those sorts of molecular questions.
Yeah, maybe I'll start there, and Shuibing, if you want to add anything. First, Nicole, really wonderful to see you. We overlapped in AMP CMD. Thanks for joining the call. And yeah, we did do some computational modeling to try to figure out what are the binding sites that are being perturbed. I completely skipped over that during the talk just for the interest of time, but it's in the manuscript. If you go look in the manuscript, we did motif inferences strategies. I don't think there's a convergence of motifs across loci. We've seen that in TTD, but again, bigger power, more signals, easier to see stuff like that. But yeah, it's in the manuscript.
Yeah, Nicole, nice to see you. I think Steve covered the question very well.
Great. Okay, last call for questions, just checking. Okay, so I guess it looks like, since this one is, all this fantastic work has sort of set the stage for now what you're doing next. And can we expect a paper soon or preprint soon coming from your collaborative group? We want to be have our hand on the wire.
No pressure, huh? Thanks, Monica.
Yeah. No, just
No, I'm just
just
kidding. Yeah, we
wishing.
have so this is kind of the initial study, right? We have a much
Yeah.
larger sample size now. The data has come back. We're QCing the data to make sure everything is high quality that we're looking at. But some of the questions that you asked where we were like limited by sample size and number of cells and nuclei, that's gotten much larger now. I guess I'll just leave it there unless Shuibing wants
Leave
to add
the
anything.
teaser. Yeah, that's great. So everyone will be looking for that information dropping soon, we hope. Yeah, exciting times with this and a great collaborative effort across some really powerful labs. So thank you so much for joining us, and we look forward to speaking with you again in the future. Thanks again. Have a great rest of your guys' day.
Yeah, thank you for hosting us, Monica.
Thanks.
Thank you
Bye-bye.
very much.